QArt Medical is proud to introduce the Q300, a revolutionary imaging technology for acquiring, measuring, and quantitatively analyzing individually selected sperm cells for ICSI procedures. With the Q300, you can provide your clients with a cutting-edge IVF service by making an informed decision regarding the sperm cells selected for injection. The Q300 workstation seamlessly integrates into your existing workflow. A semen sample, after preparation such as swim-up, PICSI, or any other method, is provided in a drop on a standard IMSI plate along with blank drops for the injected oocytes and a matrix of smaller droplets for individually storing selected sperm cells. The prepared plate is placed in the Q300 workstation. After logging in, the patient information is input into the software. Begin by selecting a sperm cell according to its motion characteristics. Set the magnification as used on the microscope, make sure the correct PVP concentration is selected, and click Motion Analysis. The system assesses the motion parameters in just a few seconds. In this case, the user wants to see the 5 cells with the best dynamics. The display is augmented with markings around the sperm cell head, the recent trajectory trail, and its motion ranking. Number 1 for the best swimmer, number 2 for the second best, and so on. The color of the surrounding box represents the motion quality group where rapid progressive is grade A, green, slow progressive is grade B, yellow, non-progressive and immodal are grades C and D respectively, red. The user may click on any tracked sperm cell to have the system individually track it with high fidelity until the user decides to select it for use or cancel tracking and return to examine other sperm cells. Once a cell is collected and placed in a clear area of the drop, the user may switch to holographic morphology analysis. Before imaging, the sperm is brought to and held in the imaging area, marked blue. When in place and in focus, imaging is initiated. A low-intensity coherent light source is turned on, and the system accumulates images until the user ends the capture. The system processes the captured sequence and provides an indication of the sperm cell's compliance with the WHO parameters. The report shows this sample complies with the WHO 2010 guidelines for sperm selection, as all measured features are within the required range. This sperm cell will be stored in drop A1. After a new cell is selected for imaging, the process repeats. This time, the selected sperm cell appears to be out of the recommended range for the WHO 2010 criteria. Therefore, the operator decides to release it and a new one is selected. When enough cells have been selected, a full final report is issued. The operator can then introduce the oocytes onto the dish and move to the ICSI station to continue the ICSI injection process. All selections are recorded and can be reviewed later for further evaluation. QArt's Q300, Seeing the Unseen.